Hi, welcome to the Mathemagic channel. Today we have a very special guest, and his name is
Dr. Habib.
Dr. Habib. Yes, so I'm
Right.
not going to call you Mr. Habib. I'll address you as Dr. Habib. And so thanks for doing this with me today. Where are you currently? Like where are you sitting right
Well,
now?
yeah, currently I'm in the Chinese University of Hong Kong.
Okay.
So it's a very specialized research environment situation called Random Show Science Building.
Mm-hmm.
Yeah, just next to my office and my laboratory where we do biomedical applications and miniature devices.
Wow,
Yeah,
that's that's
yes.
that's amazing. So, which part of Hong Kong is CUHK for those who don't know?
It's in the Shatin next
Okay.
to Mount Shan, Tolu Harbor, or just on the neighbor of the science park. Like Hong Kong, we have a two science park. One is next to CUHK, another one is in the cyberport. So it's basically on the typo side of the new territories. Yeah.
I've been to the one in Cyberport before, but I've never been to the one that you're at right
Yeah,
now.
you're
So,
definitely welcome to visit. Yeah, because Science Park and CUHK is just like a neighbor by neighbor. Yeah.
okay. Well, I mean, there's so much interesting stuff that you're doing, and so I was going to ask you first to explain a little bit about your background in terms of, maybe the early years and when you came over to Hong Kong and, your education and stuff that interested you as a young person, and because now, I mean, how old are you now?
I'm 34.
You're 34.
Yeah,
Okay, so
34.
when did you
Yeah.
first come to Hong Kong?
Well, I came to Hong Kong on 2017 in February, early
Okay,
2017. So
so
it's about 8 years plus. Yeah.
okay.
So that I'm in Hong Kong. Yeah.
So
Yeah.
so what was the journey like?
Well, I will start from the beginning. Yeah.
Sure. Yeah.
So basically, my by training or by education, you call it, I'm a electronics engineer, microelectronics engineering. So during my childhood, I was thinking if I want to be educated, I want to be, engineering field. So that was my very childhood ambition. Then I moved to during my, I think around when I was 17 or 18 in back in 2008, I got an offer from University Technology Malaysia in UTM.
Okay.
So it's in the Johor Bahru of the Malaysia next to Singapore border. So this is their top engineering university. So I got an offer to study microelectronics engineering as a bachelor program. So once I got it, I immediately accepted it, and then I flew to Malaysia to do my bachelor of engineering. Then during my, I think, third year or something like that, so I had a chance to do the internship opportunity. So that time the field of nanotech and biotech is just evolving. So and then nanorobotics, biorobotics, those things are really excited me, and then I feel like I want to in this field of the very research field that where I can do contribution to the human health or human development in general. Then I found a professor. I still connected with him, Dr. Rijuan. Now he's a professor, Rijuan, back in University of Technology Malaysia. Then I found that there's a new professor. He just had PhD from Nagoya University from Japan, and then moved to Malaysia. Then I feel like, okay, let's write to him if I can get an internship opportunity. Then I got it. Then I used to work on the biophysics, like developing the device that can, sense cells, single cells, like cells membrane, organelle, or even intracellular region. Then I got a chance. Then I did my internship, and then subsequently I did my final year project, and then right after that I'm already in full into my research. My heart and soul is
Okay.
like I am
Hang
really
on. You're
liking
going
it.
so fast. So this was all in Malaysia.
Yeah, it's all in
So
Malaysia.
this
It's
is
like
okay.
it's a beginning.
What,
It's
but what was your bachelor's of engineering? Was that mechanical, electrical? What was it?
it was in microelectronics engineering.
Microelectronics. So tiny
Yeah,
electronics for those who don't know.
tiny.
Okay. All right.
Yes, very tiny electronics. So basically making the chips, like, now the NVIDIA and then Intel, those are making the chips. So that was my first training, that how to make a chip. Mm.
Okay. Wow. That's pretty amazing. So you're in Malaysia at this point up to 2011 or what then?
Yeah, that time I'm talking about 2011 and 12.
Okay.
Yeah, that is my final year project as a bachelor program. So
So
still
what
very
was your
new.
final year project about?
Well, it was about developing a nano needle that can do
A
cell
nano
surgery.
needle. A needle.
Yes,
Okay.
yes, I still remember. We want to do surgery on a cell, like,
What?
uh,
You
yeah,
want to
just
do?
like
Oh
a,
my God.
Yes, it's like if you want to do some gene editing or study inside the cell, you need to open up the membrane. So how do you do
Yes.
that? So we want to cut through the membrane with a very, very tiny needle,
Oh my
probably
God.
opening a door to the cell. Yeah.
So can we just talk about the size of that needle?
Yeah, it's about 200 nanometer in a diameter.
So that's 10 to the negative.
9.
So it's a billionth of a
Yes,
meter?
billionths of a meter.
Oh my
Yes. Yeah.
God.
Yes. It's really a billionth of a meter.
Wow.
So
That's
that's that
incredible.
we make it like with some
How
advanced.
how do you build something like that? What kind of factory or manufacturing or what kind of
Okay.
device can make that? Like you make it under a microscope, I assume, or like how does it work?
Well, not it's a microscope, but electron microscope.
I mean, a super microscope, right? So,
Yes, a super microscope. Well, we call it electron microscope. Electron can reveal you 10 to the power negative 16 or even 19, the size of the electron.
right.
So basically, what we do, we call it focused ion beam, the technology.
Mm-hmm.
So we kind of shoot high energy electron to a needle, like a normal, like a large needle made by silicon or aluminum or even gold. So then we focused with high energy electron, so that high energy electron will cut the needle in a very, very, very fine shape.
What's the needle made of?
That time we made the first needle was made with aluminum.
So you had
Then
oh my God, it must be so fragile and so tiny.
in the,
You can't see it, right?
oh,
Like,
we can see it. We
there's
can
no
see it.
way
We can see it.
the
Oh, we need
human
to
eye
see it.
cannot see that.
Women, I we just see something, but inside the needle, well, we can't really see the
You're able to
tip of the
you
needle.
can't even hold it. It's just like it's so what this a robot that's holding the needle for you or
Yes. So we developed some robotic controller with a actuator, and then we control by electronics, like developing some voltage and current, simple programming, and then actuator will move in a very, very precise. Like we also call it PZT, if you heard of it.
no.
PZT is a piezoelectric material that
Hmm.
can generate some force by voltage. So you give 1 volt, and then PZT will move 1 micrometer, something like that.
Oh,
So it's a linear
so you're
relation.
controlling it with little handles or something or
Yeah, we control by inputting electrons like voltage.
oh my God. So
Yes,
the electrons help the needle move around in like an
yes,
X and Y axis kind of thing.
yes. You're right. You're right.
That is incredible. Oh my God.
So
So this was just your final year project. We haven't even
yeah,
gotten into what you're doing
no,
now.
no, 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 not yet. It's just my final year project. And then apparently in the university, they have an exhibition every year or some conference, something like that. I remember I got a gold medal award back in my final year project. So it was really exciting for me to keep going. And I got an offer to do my master's of engineering in research mode. Yeah. So I continued that project until I finished my master's.
So where did
And
you do
then
your masters? Also in
same
Malaysia?
university, yeah,
Okay,
same
yeah.
university. But I had an exchange program in Japan, in Nagoya University, where is next to the Nobel laureate that invented the blue LED. Just next to that lab, we call it I think the micro nano system engineering lab under Toshi Fukuda. Toshi Fukuda was I mean, I think last 2 years, he was the IEEE president worldwide. So I had a very lucky situation to kind of
He
indirectly
was the
supervise.
what president? I didn't catch that. I IEEE.
IEEE.
Okay.
So
What
is
is
the
the IEEE? Is it
it's
Institute of
Institute Electronics and Electrical Engineering.
Okay, all right. That
Yeah,
so you were did you get to meet that guy? That
yeah. He was my kind of
he was your mentor?
supervisor,
Oh my God.
supervisor, like
Okay.
grand supervisor. Yeah,
Wow,
yeah,
the grand
yeah.
supervisor. Okay,
Grand
wow,
supervisor. Yeah.
that's incredible. So so you finished in Malaysia. Did you go to so you went to Japan? So
Yes.
you were in
Went to
Japan for
Nagoya.
a while?
Yeah,
Okay.
yeah.
And then you came to Hong Kong or?
Well, then I finished my master's. Then I was looking for my PhD. So, but searching PhD is a very exciting journey. You can't just get anywhere. So you need to write to professors, and then then wait for the scholarship, and then there's a lot of process. So I find like my current supervisor that I did PhD in Hong Kong. Back then, she was in Denmark in Aarhus University.
Mm-hmm.
So in Denmark, Aarhus University, working also in the same field that I'm working. So I wrote to her, and then she was in Denmark, and then we could not really find the scholarship by that time. Then I
So
was a little
hang
bit,
on. All the
hmm?
universities that you went to, did you always have a scholarship?
Yes,
Oh my
all of
God,
them are.
that's incredible. Based on I imagine academic excellence, right? Or
Indeed. So
okay.
everything is under either scholarship or under stipend. stipend, like some sort of payment.
That's
So
wonderful. That's amazing. I mean,
mm.
you don't need the congratulations, but that is pretty amazing. Okay, so this professor was in Denmark, and then did she move to Hong Kong and get you?
Yeah. That time we couldn't find a scholarship. Then I was thinking what to do. At the same time, I got an offer from a university in my home country in Bangladesh, Dhaka. So, but the offer was interesting in computer science. So I had to teach computer science. Then I find it okay, because in the computer science, you do have a very integrated part of computer engineering. Like again, making the architecture or hardware, designing those things. There's a almost half of the computer science and engineering are focused on like how to make the chips, for example. Then I teach over there. I teach assembly language, computer architecture, and then very large scale electronic design, those things. Back in Bangladesh, I spent about 1 year, and then the professor Megan So came to Hong Kong from Denmark, and then we are still connected. Then she said that in Hong Kong she's opening her lab. So if I want to move to Hong Kong, then I feel like, oh, that's a good opportunity. Then immediately I came to Hong Kong.
And so she's still here, this lady, or?
Yeah. She's a associate dean of the Faculty of Engineering in Chinese University of Hong Kong.
Okay, so she came to CUHK, you came to CUHK, and
yes.
What did you guys start working on then?
All right. So coming to the CUHK, because by that time, then I realized that, remember that I was talking about the nanoneedle, right? So the nanoneedle, the challenge is that the throughput of the nanoneedle is very, very low. So let's say if you want to do cell surgery,
what? Sorry,
we call it throughput, meaning.
Can you spell that word?
Yeah, throughput is like not really robust. So we need to do one by one.
You mean the sturdiness of the needle?
Not a sturdiness. So let's say you want to do experiment on a cell, right? Yeah. So with one nanoneedle, probably you can do experiment with one of the cell only, right?
And then the needle will break?
Needle will break, or it's a tedious, because of course it's a very good opportunity to study fundamentals on the cell, physiology, but the issue is that in human body, we have like trillions of cells. So to get a statistical data, so you need to kind of robust the technology, right? And then you
need to be able to experiment not just on one cell, but quite a few.
Yes,
quite a few. To have good data.
Yeah, to have a good data. There comes the microfluidics. Like microfluidics enable the cell in a flow environment. It's more like a native human body, let's say in the blood flowing.
Microfluidics. So
microfluidics.
So what is this? The study of small fluids, or is it
Yes, generally you can call it a study of a small fluids, but we kind of take the advantages of the manipulation of a cell in a very, very small scale amount of fluid. The reason is, for example, we can mimic the blood flow with just 100 to 200 microliters. So to give an idea about microliters, if you think about a simple drop, let's say a drop, simple water drop or raindrop, it's about 100 to 200 microliter.
Okay, so within a drop, how many cells would you have?
Well, depend on the cell concentration, but we can have 100 to thousands of cells in a single drop. And then if you flow in a chip in a fluidic environment, then we can have the like hundreds of cells analyzed in a very, very, very short period of time, let's say a couple of seconds or even less than a minute. So there I find we can do more research, and then there is a lot of research are going, like how do you manipulate cells in a very, very small scale. We call it miniature devices. Yeah. So
how to manipulate them? Because so I was reading very briefly, and I will probably not understand most of what you're doing, but I picked up on something which said that the movement of the cell is predictable.
Yes, true.
In a microenvironment, which means that instead of it being random or chaotic, you can kind of figure out where it's going to go. So
yeah, because you control by very specialized syringe pump. We call it if you notice in the hospital, say especially in the
triage pump, like a
syringe pump. So we push the syringe pump.
Okay.
Yeah. So if you ever go to NICU, ICU, like in baby hospital, I remember when I'll talk about that, my sons, right? So when they want to inject some of the medicine to the newly infant babies, they use those syringe pump. It's like they infuse very tiny amount of fluid in a, like, let's say 100 microliter in a 1 hour. So they use a very high precise syringe pump. We use those syringe pump in our laboratory to manipulate or control the movement of the cells.
Oh, wow. Okay. So you're working on all this. My God. Can you talk to me about MEMS?
MEMS.
The MEMS, the pressure
Yeah.
stuff that you were talking about?
Yes. MEMS is the combination. Like in previously, I talked to you about nanoneedle.
So what does MEMS stand for?
It's micro electromechanical system.
Micro electromechanical system. Let me try and grab. So small mechanical electrical systems.
Yes.
Kind of.
Yes.
Okay. All right.
Yes.
What do we do with those?
I'll give an example. So I talk about nanoneedle, right? So that's a micro scale part, right? It also have an electro because we control by voltage, and then we need to have a system like a complete loop. Then how do you make a complete loop? We put it in a chip, and then with microfluidic chip, and then we can control by syringe pump. So altogether, it become a micro electromechanical system.
So you put the liquid in in a chip?
Yeah.
No.
yeah, correct. So we do have the microfluidic chips, and then inside the chip we can have different electrode sensors.
So inside a chip there's some fluid.
Just some fluid and also some electrodes
and some sensors. So the sensors are like almost like a trigger, so that you can work with that trigger electrically to produce some. I mean, I don't know what you do with them, but there's electrodes.
Like one of the recent project that I'm working. So it's like we put some, we have a micro again as a channel, and then we have an electrode in between the channel.
I hope this is not top secret, by the way.
No, it's not top secret because I already published it earlier.
Okay,
so that one. You can imagine. So electrode basically have an open loop, right? Mm-hmm. So anything in between, if you change the electrical conductivity of the medium, we can probably detect by changing of the voltage, resistance, or capacitance, right? So then we flow the water in between the electrode, and then we can know about what's going on about the water. If the water, can be blood, or can be cell as well. So that's whole together is call it a system. So that at that point we only call it a MEMS, like micro electromechanical system.
So any small system that's electrical is a
MEMS, yeah. Okay. So any small system that's electrical and also have mechanical, because we need to put pressure.
Right. Not just electrical.
Yeah. Right. Just we need to connect the fluid with syringe, and then it has to be closed loop. Otherwise, the fluid pressure will leak, right? So we need to have a closed system. So therefore we call it a MEMS or micro electromechanical system.
So I asked you a very silly question, which was, how do airbags work?
Oh yeah,
is that connected to MEMS or not?
Yes, it's definitely connected to a MEMS. So if you think about
because you have an impact and then there's pressure, right? But I don't know how small the thing, if it's behind the steering wheel or if it's in the engine. I don't even know where it's located. But there's got to be a pressure reaction that operates really fast. For the airbag to come out. So we're talking about cars right now. But how would that work?
Yeah, that's a very interesting question and very, very good application of the MEMS. If you know about airbags, right? Airbags, the concept comes like very early, I think about almost 50 or 60 years or even earlier. But it has been only implemented recently because previously the pressure was not really sensitive enough, or the equipment was not sensitive enough to, kind of blow the airbags so that it's safety.
Question. Is the airbag sensor at the front of the car?
Yeah,
because where's the impact? Like the impact is from the front, and then the car folds.
It's in the front of the car. So I will tell you.
Yeah.
But very front, I'll tell you what happened, because it has a the sensor, the triggering sensor. It has like a metal ball, like a simple like a ball, bearing ball. So the bearing ball balance based on the gravity. So it's just a simple ball that is based on the gravity. So when there's an impact, so what do you call, the ball will displaced. Whenever it displaces, it completes a circuit. So when it's complete a circuit, it generates some electricity. And interestingly, to your surprise, that airbag, the air is not really compressed air. So it's by explosion. So there's an
explosion of air.
There's an explosion. The explosion is
air. I don't know what gas fills the airbag.
It's called a sodium azote. So it's basically nitrogen and sodium. So it's in a dry environment, it looks like a salt. Is in a dry environment, it can be like, just like a salt. We can keep it under the steering wheel. And then when there is the electronic short circuit happen, when the ball moves to the
It's a very
sensor,
fast reaction then.
yeah, well, it will explode because electron moves very fast, fraction of nanosecond, very very fast. Only electron move faster than any other things, right? Electron and light. So we can only send whatever we have existence. So electron is the fastest one, possibly other than light. So and then it will explode. Once it explode, within a few milliseconds, the airbag become complete.
Wow.
Yeah. So it's not like it gives some pressure, because pressure is really
No. I
slow.
see. So you're saying
Yeah.
it's displacing a ball bearing, and that displacement closes a circuit, which sends an
Yes.
electron to
Yeah.
like open, what was it, sodium and azotate,
Sodium azode,
nitrogen,
sodium,
what was it?
yeah, NaN3. So
NaN3,
yeah,
which is in
yeah,
what, solid form?
in solid form it
Under
explode
the
and become a
steering
gas.
wheel, and then it
Yes.
explodes, and then suddenly your
Yeah.
airbag saves your
Yeah.
life.
Yes. That's how it works. It's like that's why
Oh.
you can imagine. So when if you see any of the movie or some of the videos, so it will make a sound like pop sound that airbag suddenly comes. So that's the tiny explosion that happened.
Yeah, sorry to ask you this question, which is off topic of your research, but it's just out of my own curiosity, I guess. But back to your research then. What are you working on in terms of cells? Are you still working on cells in general nowadays? Or
I do.
because you mentioned earlier, like it would be interesting to talk about cells as powerhouses. So, or is it what do you say, mitochondria?
Mitochondria. So
Okay, so
this
for
was
those viewers who are not sure about the structure of a cell, I don't want to go back to biology 101, but so inside of a cell, there are mitochondria, which are what? They're the energy sources, if I remember my biology course correctly.
Yes, you're correct. So mitochondria is called powerhouse of a cell. So it performs the metabolism, like creating the oxygen, and then it creates the energy for a cell. That's how the actually the cells get the energy to grow and divide, and then basically power the whole human body. Yeah. So this project is very interesting because this is my first project when I started my PhD. So like the concept was like that. So mitochondria has a DNA inside the DNA. So you have a nucleus, right? Nucleus has a DNA, and other than the nucleus, mitochondria is also called it as a living organism. It's like almost like a bacteria inside the cell. So you can imagine. So a cell is about 30 micrometer, 20 micrometer. So if you want to know micrometer, here is about 100 micrometer, the diameter. So a cell is about 20 to 30 micrometer. It can be smaller, but generally, let's say the stem cells or cardiac cell or any muscle cells, something like that. So inside the cell, we do have micrometer, like 1 micrometer or even sub-micrometer mitochondria. Those mitochondria is continuously working for us, and then producing the basically the power for the human body. So it worked like a. So then what we do, so we are trying to in the chip again. So we study cells, fine. Then we want to see what is the function of mitochondria in terms of their, how mitochondrial quality impact the cell's quality. So then what we do, we compare the mitochondria of different cell types. For example, we have neurons, we have muscle cells, we have stem cells, we have blood cells. Then we found, interestingly, the neuron cells, the mitochondria are much more active compared to muscle cells. So that means what? The neurons, actually, we need more energy to function the neuron, because like sending the signals, so it's continuously working. Even in the cancer cells,
Do you
the
think,
quality.
sorry to ask you, but do you think, is it because neurological activity is more complex? Almost like when you have a computer open and that you have many applications running, you need more power than if you just have one application running. Like, I'm comparing it to the muscle cell, where maybe the muscle cell is just geared for like a stretch and a contraction, whereas the neurological cell has more, I don't know, more stuff happening, more
Yes,
things
indeed.
running, or
Indeed. That's why we did not really surprised to see the outcome of the experimental
you
result. Is that
expected it? All right.
yeah, we already have some idea about the literature. That's why we bring neuron and we want to see what happened. We find that the mitochondrial concentration in the neurons are almost double compared to the cardiac cells.
Oh, I was going to ask you, like, how many times more? Like, is
Yeah,
it, let's say,
almost
is it like
double.
1% or 2%? But you're saying like, so the mitochondrial energy
concentration.
output. Is it
Yeah.
energy output, would you say?
We call it a concentration. It can be correlated with the energy output. So it's like number of mitochondria in a cell is almost double
Or
compared
the
to the,
number of so there's not just
yeah.
one mitochondria per cell.
It's not mitochondria per
So
cell. It's
just
many.
to give an idea, like in one muscle cell, how many mitochondria do we have?
It could be a lot actually. So it could be lot. Yeah. So it could be millions of mitochondria.
Millions in
Yeah.
one cell.
One cell. So
And so you're
in
saying if we
my,
have millions in one cell, then in a neural cell we will have like
yes.
2 million,
Yeah,
twice
something.
as many.
Yeah, it will be because the neurons are bigger.
Oh my
So
god.
like it can be as big as like
Oh,
connect
are you
very long.
saying the neurological cells are in size bigger than the muscle cells? They
Yes,
have
and
more
also
space?
more space, and also they need to be more active. Like they have dendrites, nucleus, and also axons. So everywhere we have mitochondria or more
Wow.
mitochondria there.
Oh,
Yeah.
that's super interesting. How do you study those things? Like, what are you observing? Are you
Well,
prodding, pushing,
yeah.
moving stuff around? What are you doing?
Oh, that's a good question, because mitochondria is so small that we can't really see under normal microscope. So we need to go to super resolution microscope, and then the probing become very, very limited with super resolution microscope. We cannot just bring a nanoneedle inside the super resolution microscope to probe. Then what we do, we chemically quantify that. So
Chemically, okay.
we do have some chemical sensor that chemical, basically it eliminate fluorescence. So based on the activity of the mitochondria, so we can detect those fluorescence by confocal microscopy, if you ever heard of it,
Can
or
you
the
say that again?
confocal microscopy.
Confocal.
Yeah,
So
or flow.
CON focal.
Yeah, CON focal
So
microscopy.
with focus. I'm trying to
Uh,
resort to Latin here. Confocal.
yeah, confocal it only detect the fluorescence signal based on the layer. You can imagine. So let's say if you put a light like in my camera, so we have a different layer, right? Like it's showing me here. So it's tiny, like millions of layers, for example, that there. So the confocal allow you to get the signal using a pinhole from a very, very dedicated amount of layer that you can define.
Wow.
So then we define it. Okay, give me the signal from 10 micrometer. Then whatever fluorescence we have, we can only get it from that tiny bit of 10 micrometer in the layer, not just on the surface, but also layer by layer. So it's like manipulating the focus point in a when we have a, how to say, when light path, right? You can imagine light path where the light are going. So we can cut the light path, and we only can detect from a very specific point of the light path.
So it's so interesting that when you look at very small things like that, the way to approach the situation has to change. You're
Yes.
talking about like disturbing them, having a look at them with chemical fluorescence, and
Yes.
it's so different to what our perception of the world is, because you're operating on such a small level. So is this what you're doing nowadays? This is what you're working on these days?
Well,
what's your current area of interest, like right now?
Okay.
Now, like
So,
what are you doing today, for example?
okay, today I'm working on currently today I'm on okay, I'll talk about another equipment, then you will know what I'm talking.
Okay.
So you may heard of another equipment. I know that I'm giving you a lot of
Well, please don't assume, because
buzzword.
I know nothing about anything. This is all
Okay.
like, for me, it's a wonderful discovery talking to you right now.
Okay, there's a thing called flow cytometer.
Okay, so fluo cytometer.
Meter.
So let
Flow
me think
flow
about
cytometer.
flow.
Yeah, flow cytometer.
So FLO, and then cyto, I imagine that's CYTO.
Yes,
Because
and the
we're
meter.
talking about that's the word I'm catching, and meter, okay, measuring kind of.
Yeah, measuring. Cyto is referring to cells.
Yes. So
it's like a measure cells in a flow condition.
okay. So it's as they move around, kind of thing. Again,
Yeah, as they move
that
around.
goes back to your microliquids, right?
Yeah, we have a $1 million equipment, flow cytometer, already in the market. So what it does, we modify that flow cytometer to make it pushing the boundary of the flow cytometer application. So what it does, you sort the cell based on the fluorescence signal again. So I'll give you an example. Let's say you want to understand some of the bacteria, or forget about bacteria, some of the chemical presence in a cell or not. Okay, so then what we do is stain the cell with some fluorescence molecule. So if you get a positive signal, for example, a disease marker from that cell, you'll light up with fluorescence, right? That fluorescence can be detected by, photodiode or laser sensor inside the flow cytometer. So whenever it detects, it pushes the cell or sort the cell, like separate the cell based on the
How does it
fluorescence.
push it? What kind of things does it have inside that it can push them and pull them? Like it's
Oh yeah, there are many things. Easily we call it DEP, dielectrophoresis. Another another
oh, okay.
electronics. Yeah, so it's like a putting an electrical charge in a fluid environment. So you'll move the fluid, and fluid will move along with the cell.
So you see, like when we think about sorting in real life, we think about maybe like trap systems or pushing systems. But when we're looking at it, so now these are like electron systems that are pushing
Yes,
and pulling
yes,
because it's
yes.
so
Often,
small.
often we do is like we encapsulate the cell in a droplet. You can imagine, like put it in a small droplet, and the droplet is electrical charge sensitive. Then if you charge it, and the droplet will move, the droplet will move along with the cells. That's
Okay.
the function of the
I'm
flow
going to ask
cytometer.
you a question. I don't know if this is ethical or not. Where'd you get the samples from? Like, where would you get a droplet of neuro cells? How'd you get that? And how'd you
Oh,
get a droplet of muscle cells? Is that from a
yeah, yeah.
human body, from an animal, or what are we looking at?
Well, it has to be go through the hospital authority approval, Hong Kong.
Okay.
Yeah. Then we do from animal, from mice.
Okay. So from
Yeah,
mice. So you're
from
studying.
mice. Yeah.
Okay. So if you want like a neuro cell sample, like a one droplet, what is it? It's a drop of
Yeah.
mice
So,
brain liquid, or
well, no, no. So, well, again, when you talk about this, because every step has a lot of detail, so I'll try to minimize it. So when you talk about cells, we do have two types of cell. One we call primary, another we call culture. Primary means that you do the surgery from the cell, you take out the neuron, and then you grow the cell. That's we call it primary, directly collecting from the element. Another one, you can buy the cell, like we call it a passage. Like you buy it from ATCC. ATCC stands for some of the organization that kind of control or regulate the cell growing. So you buy it from the library or cell bank basically, and then you bring in our laboratory. We do have right here. So we have an incubator to grow the cell. We can grow and multiply them, and then we do those cells as experiment.
so how do you grow a cell?
Oh, in an incubator. We do have an incubator,
So
cell
what's in
growing.
the incubator? Like you open the door, it's like a sterile environment, and there's
Yeah,
like a little petri dish or something, or
yes, yes, you got it right. The difference is that in the incubator, it maintains the sterile and it maintains the carbon dioxide, oxygen, and pH level at
so
all
the
times.
air levels around it.
Yes,
Okay,
yes. That's the difference
and
of the
temperature.
normal.
Yeah.
Yeah. So that's a controlled, basically mimicking more like, healthy or happy environment for cells to grow. It's like a warm, little bit warm, 37 degrees Celsius.
A happy environment for cells.
Yeah, we call it happy because we want to protect any contamination, bacterial or
Sure.
any other contamination. So, and then we let them grow in a petri dish, and we change the medium, like growing medium. We have a highly nutritious and a well growing medium, some antibiotic. So we culture them. Let's say every couple of days we change it, and then cell will grow. Then we separate them, and then we can count using the hemocytometer. Uh,
Okay. That's
so
another
there.
machine. You can count the cells afterwards, right?
Yeah, a flow cytometer can also do that. Flow cytometer is a very advanced. I like flow cytometer because it's my ambitions in future. Actually, that's where I'm going. So
Do you
it's
know,
a
this is what I was going to ask you, is what are you going to do next? So very good segue from you to take us to your future ambitions. So yeah,
yes,
what are you doing next? I mean, you're
yes,
doing all
yes,
this work
yes.
on cells. Where are you going?
Yeah. Okay. So here I want to mention one thing is also important. That's kind of trigger my motivation. It's like, remember I told you just now that cell sorting, that we need to light up by fluorescence to trigger the sorting. But you can imagine, I think you asked this question. So if you add chemical to the cell to get a fluorescence, you are already compromising the cell, right? You can't reuse it. So there's a one more parameter, we call it biophysics or phenotyping. Basically it's like or physical parameters of a cell. So it's not like it's like you don't really we also call it a label free. So you don't really label with some chemical, but what we do, you give some mechanical pressure on the cell, and then you observe the response of the cells. For example, we also call it a deformability. So we deform the cell will be squeezed, and then we observe how quickly it come back to the original shape. So that's
Oh,
referring to the deformability. So interestingly, this deformability also become a very important marker of the cell's quality. For
okay.
example, in a COVID patient, one of the recent paper from a group from Germany, so they reported that the blood cell that from COVID patient become like COVID recovered patient is much less deformable compared to the healthy or never been COVID. Yeah, so that means some of the blood cell kind of permanently become damaged. So this could be a potential long-term effect of the COVID infected patients, for example.
Mm.
And then cancer cells, those cancer cells are highly deformable. That means malleable. They can easily migrate compared to the less malleable one. So that's a biophysical parameters is a
So when you say
without
they
any
can migrate, you mean they can go to different areas of the body,
very quickly cancer cells
which
they
is
are
why cancer goes through the body
yes
faster
yes yes
than
faster than
other infections, or
other infections. So and they're very deformable compared to other
they're
cells.
very
They're very soft,
deformable, right?
deformable. Yeah. So now coming back to the flow cytometry, right? So we can sort by fluorescence, but the fluorescence is already compromising the cell. But if I study the deformability or mechanical phenotyping, we are not compromising the cells,
So you
right?
study fluorescence and deformability, like how malleable a cell is.
How malleable is that?
Right.
Then I'm modifying that flow cytometer to sort the cell based on their deformability.
Wow.
Mm.
Can I ask you two questions which might be more relevant to the general public?
Mm.
Okay, so number one, what do you think? Did you think the COVID vaccine was effective?
Well, yeah, definitely. Yeah, it was effective. Yeah, because it's a trained human body to trigger the antibiotic you need when you have a possible infections. Yeah.
Mm-hmm. Okay.
So vaccine is like a training your immune system. So just to basically give an idea. So the vaccines mean it give you some of the very very small amount
Mm-hmm.
of doses
It
we call it, right?
microdoses you, right?
In microdoses, so that your immune system has a memory. So immune cells, they know what to do once you have next time you have a large infection. So they will immediately trigger the antigen needed for the fighting against the COVID. Yeah.
Okay.
So definitely it's helping.
And where do you think COVID is today? Like I see these articles circulating about new COVID variants in the US, and then people aren't
Well,
taking it seriously anymore. So I'm
yeah,
trying.
it's not a these are not really novel. So like it's largely referring to the infections. So we could have a different type of infection. The problem is if the infections are really really brand new that we never know. So it's very hard to identify what is the type of antigen, what is the type of antibiotic, and most of the time you notice the treatment coming from the hospital is like treating the symptoms, not really treating the virus.
Mm-hmm.
Right, if I have a fever, they'll give you Panadol to reduce the fever. If I have a breathing difficulty, so then I'll take support from the respiratory system. So, and then let the immune system to generate the antigen needed or antibiotics in the body,
Oh,
right,
so you're
to fight
saying
it.
that typically speaking, we treat the little issues like the breathing issues or the fever
Yes,
issues, but your body
yes,
itself is just
yes.
fighting the
Yeah, just
antigens from your
fighting.
body. It's just fighting by itself.
It's just fighting by itself. So we only treat the symptoms.
Right.
We don't
I understand that.
we can't really alter the we can't really well we don't have it until now. So we can't really alter the
That's
disease just right away.
And
Yes.
how close are we to finding,
Oh.
treatments for late stage cancers?
Well, the cancers treatment are because cancers is very, very what do you call? Well, it's difficult in the late stage. It's the very, very. One of my project was also on the cancer, so I work on the oncologist actually
Mm-hmm.
to treat leukemia. So it's in the late stage it's become very difficult because it's spread again in the cancer. You know what we do in the cancer treatment most of the time we fight against the proteins or symptoms,
Mm-hmm.
right? So then we try to like one of my previous another project that I was working with treating leukemia again with a chip, right? Then what we do in cancer, that cancer cells again they need a lot of energy, like they need to grow faster, so they need more protein. So what we do, we kind of
So hang
suppress the
on. Do
protein.
cancer cells also have mitochondria?
Yes, they do have
So
mitochondria.
which I mean, it's obvious
Yeah.
to you, but they
Yes. Yes.
and are they more mitochondria than the neuron
uh,
cells, or is it like closer
mm,
to a muscle cell? Or
Well, it's hard to directly say, but
but you said they consume a lot of energy.
Yeah, they consume, but again, when you talk to the cancer cell, it's hard to compare with any other type of cells,
Because
right?
what, they're altering like a healthy cell, right?
Yes, they're genetically mutated healthy cells becoming cancer cells,
Yeah.
right? So when you talk about cancer cell, you may compare the cancer to cancer, but if you compare from cancer to healthy cells, it's really not very comparable in terms of the
So it would be
mitochondria.
like the same as the original cell, just mutated into
Yeah, it's
a
like growing a lot without any control, right? Yeah, so that's
or
we
like
call it a cancer.
uncontrollably dividing, you mean?
Uncontrollably dividing. It's not getting bigger, but it's getting more.
Right, right.
Yeah,
It's spreading,
yeah.
as we
So
say, the
it's
cancer.
spreading is very quickly. So then what we do, so we kind of keep the protein level lower. Like that's what you do chemotherapy, for example. So we kind of use some light and then again with some chemical signaling molecules to keep the protein level low so that the cancer cannot grow that much.
Oh, so what you're saying is we're stopping the cancer cell from doing mitosis on its own.
Yes, yes, we call it starving them for, a kind of
So we starve the cancer cell from
we
protein.
starve them so that they can grow. That's why the side effect come. If you notice any of the cancer chemotherapy patient
The
receiver, they
hair loss
often
and yeah.
hair loss and something. So that's a side effect. But generally, we
Because we
can't
need
really.
protein for the scalp, for the hair, for
Yeah, we need the protein for the hair, scalp, and also for normal growing, right? So that's why it is a very very critical, treatment that maintaining the level. So the physician or doctor has to be continuously tune the parameter needed
Do you
for a
know what
particular
causes
patient.
the heavy nausea when people take chemotherapy?
Well, it can be also the side effect can report to few of the, including the nausea or vomiting or hair loss or even dizziness. Yeah.
But
So,
what causes it? Do you know?
well, I can't really explicitly say, but I can assume some of the gland that produce the some signaling molecules that may be malfunction or some produce some signal that normal healthy person don't have. So, but I don't have right answer at this point, like
Okay.
the reason. Yeah.
Okay. So, well, look, I mean, this is a lot, so I'm not going to ask you any more. Too many
Mhm.
technical questions. I think for me this was great.
Well, I'm not sure how convincing because by training I'm just an engineer, right? I'm not a doctor or biologist, so I make the machines and equipment behind the treatment, right?
Yeah, I understand, but
Yeah,
I
you
mean,
can assume.
you're not posing as a doctor either. That's not your
No,
mission
no.
statement. Your mission
Yeah.
statement is in the study of the cell itself, at in
Yes.
as per your description just now. So, I was asking you questions which are slightly off topic, just out of pure interest. Just like the airbag question has nothing to do with what you're doing,
Yeah,
but
yeah, yeah.
it's just I'm trying
Yeah,
to
sure,
relate
sure.
it to what people might be interested in as well.
Yes.
But yeah, you're a dad too. So what's that like? So you have a son who's
Yes. Oh, yeah. I had
how old is
a.
your son?
He's turning to 5 soon.
Five
Yeah.
soon. So
This.
how's that experience been for you?
Oh, that's a.
How busy are you at work, and how much time can you spend back home then?
Well, yeah. That's a. I mean, I became dead during my last year of my PhD. You
Mm-hmm.
can assume like how much pressure that I need to go through during the last year of my PhD,
Sure.
and then and my son was a very very premature baby, so his birth weight is about only 900 gram.
Mm-hmm.
900 gram is really really considered very early, and he born about 27th week. Normally, a baby healthy baby born after 32 weeks of the pregnancy,
So
but
about a
he
month and a half early, would you say, or two
yes,
months?
almost 2 months, almost 2 months, two
Mm-hmm.
months passed. So then it was hard time. So he born in Hong Kong in Queen Elizabeth. So he
Did
had
you have
to
to stay
grad
in an incubator as well?
for yeah, almost he spent almost 2 months. Difficulty with breathing system,
Mm-hmm.
difficulty with nervous system, difficulty with overall weight, and then the other issues. So there I saw the applications of the MEMS again. So
Mm-hmm.
how these incubator are controlled, including the pressure, the ventilator, and everything. So they all coming together in a small system. So yeah, it was a very busy time back that time, but yeah, I managed. But right now situations are much much stable. He
Mm-hmm.
he goes to the school and then playing around, and I think it's the best experience of my life is being the dad and then
For
seeing how.
sure.
Yeah, so it's really different, and then I really enjoy talking to him, like trying to translate the knowledge. And actually, the best thing about one thing I I recently experienced this. So he keep asking why, and then every time I come to my office, because I work in the clean room. Do you know what is clean room?
Mm-mm.
Okay, clean room is the environment that we maintain the dust in the air.
I
So
mean, I'm
remember that.
imagining it's not dirty. That's
Yeah,
why,
no, it's not just dirty. So we maintain the environment so that we call it 100 class. So in a square micrometer area, so we keep the mimic of the particle around 100 or 100 square, something like that. So the reason is that we make the miniature devices. So those are really dust, attractive. So any dust can get easily adhere on the surface. So we environment it. And then we work under a yellow light, so you block all the UV and everything, so it's a very controlled environment. So there you need to wear a specialized dress, like personal protective equipment. So every time I go to clean room, he'll give me a call on the FaceTime, and then I want to see, okay, why do I wear this? What do I do?
what
What
are
do
you
I do?
doing?
Yeah, what are I doing?
Is it
What
like a
experiment?
hazmat suit or not? No.
Huh?
Are you
Huh?
wearing like the same as the hazmat suit we had during COVID?
Yeah,
Or
yes, exactly. Almost similar, almost very similar. So yeah, and also some of the chemical we work are very sensitive to the breathing system, so we block out that, and then that's how we call it a clean room. So very special environment. So my son looks like very excited
he
whenever he
doesn't know
sees in
what's
the clean
happening
room.
in the clean room.
Yeah, he doesn't know what is
What's
happening, so he wants to go there. I say, okay, you want to come here? All right. So yeah, so that's
Do
like
you know,
a
I still remember during COVID, people at the airport in hazmat suits.
yes,
They were at the
yes,
airport about to get on the plane and they just don't want to
yeah,
get an infection, so they were ready to
yes,
fall on a
indeed.
hazmat suit.
We
Yeah.
we do we do wear it every day.
No, no,
Yeah.
I imagine you don't go to 7-Eleven or the supermarket with your hazmat
Of
suit.
course not. Of course not. Yeah.
Do you think, by the way, that masks were effective back in those days?
Yes. Actually, what mask does, you need to think a little bit about the microfluidic again. So when you breathe out, we just simply don't breathe out carbon dioxide. We also breathe, or kind of when you speak, right? Some of the fluid droplet come out.
Hmm.
Those droplets are dangerous compared to the air. So mask actually block those droplets.
So you're saying that the droplets, which so the humidity
Yes,
contains
yes.
viral
Yes, viral
um,
or infection or any other contamination that can
it
cause
can.
the disease. So masks actually block out the droplets.
That's interesting. Yeah.
Mm-hmm.
Okay. And so you said you're waiting to go to the States. So this is a conversation you and I have been having recently about you waiting for a visa. And so what's happening with that? What's
All right. So talking about the flow cytometry back again. So I found a
which
group.
university would you go to? Like, what was the plan?
Oh well, my offer is in from Qualcomm Institute. So
Where's Qualcomm?
Qualcomm is a independent research institute in University of California, San Diego.
So this would be like a private company hiring you. This would not be. Would it be
Well,
university funded
yeah,
or?
it's a university. So it's like this. So like all those major, electronics or in your founder, for example, Qualcomm, Facebook, or even IBM and those things, they have some sort of research center in many of the universities. So those research center function independently,
Mm-hmm.
but heavily funded by the company itself. So I can imagine. So what it does actually, Qualcomm originally from San Diego, so they have everything is in San Diego, like the first factory and everything. So they found a research institute inside the university, and then they provide the funding to have the facility of the clean room, making the lab and everything. And then those lab actually do some servicing, like provide servings to other lab. For example, those are need of clean room facility or making the chip and device, so they need to pay, or even servicing to the startup companies, right? So it's a research institute. So I got hired in that research institute, yeah, under the UC San Diego. Yeah.
Oh wow, that's amazing. And so I what's the holdup with the visa?
Well, yeah, because I assume the what I call the visa has a well, they call it a, because my background is a little bit complex, so microelectronics, semiconductor, some of the biomedical applications, these are making I think visa, I mean the consulate officer, make it difficult to make immediate decision, like how the transition are really there. So they want to kind of, they call it a administrative processing. So they will send my information to the USCIS, and then when we have the clearance, then they can issue the visa. In my previously, I did also apply, for US visa. I got my clearance within a month. But that time to go to attend a conference in University of Chicago. But this time I'm going there as an exchange scholar. So the processing time may be longer. But I hope that will be solving as soon as possible. Then probably I will be moving to San Diego.
Do you think it's to do with the current administration, or would it have been the same 5 years ago?
No, I think it could be due to the current administration because of the uncertainty and changes of the policies. So the U.S. consulate immediately cannot make a decision. Yeah, so that's why. So they, whenever they cannot make an immediate decision, they will hold it up.
You don't seem too stressed out about it.
Well, I was stressed. I was.
You seem okay. Like you're staying at CUHK until like they say you can go. Kind of.
Well, that's a very good thing from the UC San Diego because University of California, all the University of California, including some of the top universities in Boston, they are under a program called Postdoctoral Association. So the Postdoctoral Association claims that when a university gives an offer, the offer is valid until the candidate don't cancel it or the US visa don't reject it. So the professor that hiring me, he told me you don't have to worry. So the offer is valid even for years. So if you get a delayed response, we are fine. So you will be just joining as a delayed. So you don't have to worry about that the offer is gone. So that's why probably you may find me a little bit relaxed. Yeah.
Yes, because I was just kind of wondering, like, this thing's not getting approved. You're sitting around waiting. And it's like,
it's really stressful.
It's a standing offer because, I mean, for someone of your caliber, obviously it's...
Indeed. It's like because now my everything is hinges on that, right? So talking about that, like, well, that will be my before, like the postdoc is for 2 more years. So after the 2 years, then I shall be able to apply as a tenure track assistant professor position. So that tenure track position is really hinges on my postdoctoral training. So I really need to have that 2 years training, and then I can apply for tenure track position either in Hong Kong or any other country.
Tenure, right?
Yeah, tenure track. Okay, so it's like a assistant professor.
So to have a professorship.
Yes.
Okay, and then you'll be teaching at university, I guess.
Yeah, in the teaching and then doing research. Yeah.
Fabulous. My God.
Yeah, that's my ultimate.
I know this is where you're going. Right? I've had the opportunity of having you work for Mathemagic as well, which has been quite wonderful. Everybody's been very happy to have you come and teach their kids. I don't know if you have anything to say about that to parents who've worked with you or to parents who may want to work with you while you're here. Or in terms of your experience with tutoring, just to wrap up.
Well, yeah, that's a very wonderful experience working along with and Mathemagic, and also especially giving me the opportunity to expose, and I call it different education curriculums. For example, I was teaching especially on the creative math learning, for example, from material from Beast Academy, John Hopkins youth program that I was teaching to Nathan. Now I am still connected with their parents sometime. So I remember, I think Nathan is in Georgia Tech right now. And then another one from HKIS. And then and then what else? I forgot his name, but it is so like I have been teaching multiple child from a single family, right? So it's really exciting journey. So I can see that it's very satisfactory that that younger son is, like, for example, enjoying learning with me, and then the what do you call, the next one is coming up again.
And then they'll say, "Have you teach my younger child?" Because
yes,
Georgia Tech.
Yeah, that's really exciting journey for me, and I like teaching. My mother is a teacher.
Okay.
Yeah, so I enjoy transferring my knowledge. And then I think you may notice some of the, I'm not sure if you ever get a feedback from the parents. So I don't really teach the normal way of learning the math. I try to find other way, meaning the creative way. So using the imagination, not by pen and paper, using the imagination, how to solve the mathematical problems. And then I call it a physical meaning of the math. Right.
Physical meaning of mathematics. Okay.
Mathematics, right? So that's my ultimate theme when I teach something. So let's say, like, you find a square root, right? Then what is the meaning of that square root? Like, how do you physically anticipate it in a picture or a diagram? Or if I give you a pen and paper, then how can we translate that in a knowledge, right? And then dividing or multiplying or even 1/3 of it. So can it be done by imagination, just by drawing? If you can imagine, mathematics become like a given, right? So that's that's why I give emphasize that is it my student can imagine those numbers.
Yeah, I think you're touching on a very important topic now, which is if you've looked at some of the areas of research in education that apparently we're all visual learners. So even those of us who think that we're fine just doing it without any visual representation, whatever, and I certainly never thought I needed visual representations. I was just happy
With numbers.
just doing
You need to be you need to be genius for
No.
that numbers.
No,
Like
I mean,
not
just,
really.
but I think, and when I look at my wife, she needs visual representations, or my kids. Like, as soon as while you're explaining something to someone, there's moments where they kind of glazed over, like there's not really a reaction, and then you bring the visual representation and you see the reaction, like there's this little forward movement, like, and so that this is the moment where the brain is pinged. The brain is just connected to something, because it's important to try and read your student as well, right?
Yes.
Yeah, I really like what you said about visual representations. It's all the fad nowadays about how kids are learning. So
and I was excited to learn that. I remember some of the feedback, especially for Nathan, if you remember, that went to John Hopkins, right? And then he moved to Perry School. After that, his mom texted me. The teacher from the Perry School asked her, where did he learn to solve the math problem? This is like, this is so different compared to other kids. And in the Perry School is one of the top boarding school in the US. So, and she texted me, hey Habib, we want to thank you again. So it's like he's getting noticed in the school that from where he learned to solve math problem, like slightly different compared to other students. So yeah, I find it like I teach him for a long time, more than a year, I think, almost. So it's over the long period of translation of
then he gets to have so the thing is, when you have that length of time with a student, they really have a method and a technique which is imprinted by the mentor.
Yes.
So then you can really look at it like mentorship rather than just you spending a few lessons with a student. So they've really taken something from you. There's a piece of you in there. Okay, well, I guess this is over time for I've asked you too many questions, or I've
I almost forgot the time. It's really enjoying talking to you.
No, this is great. Thank you for your time. I'm sure in your busy day. Do you have any closing thoughts, or should we just wrap up here, or what's the
closing thoughts? I wish very good luck to Mathemagic. So I see that with your effort and opportunities available in Hong Kong, so you're growing, and then the center, including the YouTube channel, everything is growing really good. So I feel proud to be part of Mathemagic. Actually,
I have a book coming out, and that's
wonderful.
what I'm going to be pushing next. So
wonderful.
But yeah, the YouTube channel, I'm definitely trying to put out a lot of material and some shorts and things. So but okay. So listen, I've learned a lot today. I hope our audience has learned a lot too. I'm going to wish you a wonderful afternoon, and thank you for your time.
Thank you too.
That's it.
Wish you good luck.
Thank you, Dr. Habib.
Welcome, most welcome, Karim. Bye bye.
See you next time. Bye bye.
See you next time. Bye bye.